So let's begin again. It couldn't give me more pleasure to introduce Mike Shelley. So let's get
No, it's a super title
Medical
subtitle.
with Mike.
Yeah. Thank you, Neil. And thank you to all of the organizers. It's been a great meeting. So I'm going to tell you about some work that my group, I should say, those of you who know me, I'm part-time at the Simons Foundation now at the Flatiron Institute, which is a new research institute being developed there. I'm running the biophysical modeling group. I have a name. Okay. Not referring to myself there. So I'm going to tell you about work that my group at Flatiron has been doing with an experimental group, a biophysics group at Harvard, run by Dan Needleman. The work I'll talk about today is mostly done in collaboration with Dan and a graduate student of his, Haiyan Wu. So I'm going to tell you a bit about this structure. So this is a electron micrograph that is a reconstruction of a mitotic spindle in a single cell C. elegans embryo. And this is, I think, somewhere on the order from this center to that center, about 15 microns, maybe a little bit more, something like that. And so what you're seeing, this is really just a snapshot that's taken in some part of the cell cycle when it's going to the first cell division. And so what you see here are all these green things. There was a talk about these yesterday. These are microtubules, which are rather stiff biopolymers that do a lot of work inside of the cell in collaboration with their associated motor proteins. And there's a nucleating site. There's two of them here. They're called centrosomes. And so there's this huge cloud of microtubules emanating that have nucleated and polymerized out of this site, which is buried in the graphics here. And so there's this kind of American football-shaped object in the middle, which is a bunch of aligned microtubules. And caught here in the middle are the chromosomes. And these chromosomes, there's actually pairs of chromosomes in here, duplicates, and they're about to be separated. And this whole object, this spindle, will elongate. And the chromosomes will separate along with that elongation. So that was just, so this is actually from a paper of ours in collaboration with another experimental group in Dresden under Thomas Müller-Reichert, who does these beautiful reconstructions. What we did is we helped them understand, or helped ourselves understand, the way microtubules from these organizing centers are actually coupling onto the chromosomes and how they're connected into the rest of the spindle network. You see all of these microtubules, but only there's a couple thousand of them on each side, but only a couple of hundred of them are actually connected onto the chromosomes. So we were investigating the nature of that connectivity into this whole thing. So I'm not going to talk about that. This is really just to show you a glorious picture and give you a sense of the landscape. So microtubules and motor proteins, microtubules in particular, are one of the elements of what's called the cytoskeleton to the cell, which are biopolymers of various sorts, and there's motor proteins and crosslinkers that act upon them, allow them to interact with each other and with things that are suspended inside of the cell. The kind of dynamics that you see, these elements of the cytoskeleton exercise, are kind of foundational examples when people talk about active matter as a field. These are motor proteins are consuming a fuel in order to perform work on something to which they are attached. And then it makes all of this move around. So these are all examples of that. And each of those is involving microtubules, which are these long, stiff, just like this, these stiff biopolymers, where they are put in situations outside of the cell so they can be studied in a more reduced quantitative environment. You can probe them much more easily. And so in Dan's lab, they've done a lot of very beautiful work on looking at the self-assembly of the spindle in cell extracts, where they stimulate production of eggs in a frog, give it hormones, produces many more eggs than usual. You harvest these eggs, you grind them up, you centrifuge, and you pull out the extract of the cell, which doesn't contain any membranes, any large things at all. And if you throw in a bit of DNA, any sort of DNA, could be Neil's DNA, coated onto a little bead, this object will self-assemble itself out of the constituents of this extract. And this can be very stable. It will live, so to speak, if that's the right verb. It will exist in this state for hours and even a couple of days. So it's a very stable kind of mechanical object, if you like. And it's made up of many thousands of microtubules. Now here, there's no explicit kind of organizing centers. There is a piece of DNA there in the middle that is involved. So Dan's group and work we've done with them have looked at what happens when you take this extract and you play games with it. For example, in this case, you take the extract and you throw in toluene. Toluene, that's the wrong word. Don't use toluene. This experiment will not work. Taxol. I don't know where toluene came from. I remember its smell from high school chemistry class, though. Anyway, Taxol is a anti-cancer agent that causes the nucleation of microtubules out of the monomers, out of tubulin monomers. And so what you'll form is a rapidly proliferating gel of a bunch of short microtubules. They're stabilized, so they kind of grow to a certain length and then stay there. And what you find is that when you do that, then these gels are contractile, and it's driven by activity of motor proteins that are moving upon this kind of newly formed scaffold. And this is a movie from Zonomir Dojic, where looking at a similar system where these are more synthetic microtubule motor protein suspensions. I think many of us have heard about these experiments in various ways. But this is a suspension of kind of filaments or bundles of microtubules, many thousands of them. There they are right there, kind of roiling around. So you have some sort of actively driven system that looks kind of turbulent. And what Zonomir does in this case, and I think it's a really beautiful experiment, is he elucidates through an experiment a very beautiful instability calculation that was first done by Sriram Ramaswamy and Simha, where they had a prediction that if you had a suspension of swimmers, microscopic swimmers, and the question was, could you have a long-range orientationally ordered state? Could you have huge flocks out of that? And the answer from his calculation was no, that there were instabilities that arose because of kind of transverse growth of deflections from this aligned direction. And it turns out that swimming is not so important to that calculation. What's really important is the fact that there are extensile flows that are produced by the activity of the inside of these rods. So it's kind of stretching fluid out along these tube directions. And so what he does, yeah, I can tell I'm in a real talking mood today. I should be on slide 4 or 5. But anyway, this is just beautiful. But what he does is he shear lines it. And then when he shear lines it, he lines up all these tubes, and he sees this transverse instability just pop right out. It's really the first, I think, experimental evidence that this is really that this is a bona fide instability outside of our codes and our pieces of paper. And this is another experiment of Dan's looking at the process known as polarity sorting, where motor proteins know about the polarity of microtubules. They have plus ends and minus ends. And what they do is that if you have multi-headed kinesin motors, which were also mentioned yesterday, they can attach onto two anti-aligned microtubules and flux them past each other because their motor heads are walking in opposite directions. And this is actually showing that through a photo-bleaching experiment. That spreading of that black line is the spreading of a photo-bleached region of this material, if it will do it again. And then I really better move on. You're getting the whole world of active matter and microtubules in one slide. Okay, it seems resistant to my, there it is, yeah, okay. Okay, so let me tell you a little bit about microtubules. So the first thing to know about microtubules is that within the cell they are tremendously dynamic. In many of the experiments, you use what are called stabilized microtubules. So they just come with a fixed length. They may be polydispersed, whatever, but they're just little sticky objects. And that sticky doesn't mean they stick to each other. They're stick-like. But in real cells, they are constantly polymerizing and depolymerizing in a process first identified by Tim Mitchison called dynamic instability. And so they will polymerize, and then suddenly they will switch their state from, oh, that's depolymerize. They'll polymerize, and suddenly they'll switch their state, and they'll rapidly depolymerize, so disassemble. And the typical lifetime of a microtubule inside the cell is somewhere between 30 seconds, 60 seconds, in some situations down to 10. So they are extremely transitory objects, but nonetheless, they are involved in doing a lot of the basic force transduction that's going on inside of the cell. So that's an interesting aspect. And so in that previous page, in that spindle that I showed you, that extract spindle, it's lasting for hours and hours, but the things that make it up, that give it a robust shape and form, you can probe it in various ways, is things that last for 30 seconds. Okay, so one thing that lets microtubules interact with each other are motor proteins. I've mentioned polarity sorting here is kind of a schematic of how that would work. This is associated with kinesin. Kinesin can have multiple motor heads attached by a tether, and they will walk towards the plus ends of microtubules. So if I have two microtubules that are aligned with each other, that will cause them to flux past each other. Dyneins motor a lot of things inside of the cell. It's a minus end directed motor protein. It can cause the clustering of minus ends together. We believe that that is the process by which you get these contractile gels. We have modeling papers about that if you'd like to take a look. They can transport cargoes along microtubules. They can be attached to organelles and drag them around. If you're dragging a cargo along a microtubule, that means that you're exerting a force on the microtubule and equal and opposite force on the fluid in which it's suspended. So there are ways of exerting forces on both fluids and on things to which these microtubules might be attached. It's also the case that dynein can be bound to the cortex or to the periphery of the cell and actually pull upon microtubules and apparently depolymerize them simultaneously. So they can be pullers that are kind of pulling it, yanking at things. So I'm mostly going to be talking about dynein today. And I'm going to talk about it in the context of some transport processes that go on in the lead up to the first cell division. And this is a problem I've been working on for a while, but I think we finally have a good handle with aid of some really beautiful experiments from Dan of what's going on. And so what's being shown here is a movie. And the tubulin in this is fluorescing in green. And the chromosomes are in red. Those are what are called the male and female pronuclei. This is right after the male has inseminated this egg, and the male and female pronuclei have come together, and this kind of combined object needs to make its way into the center of the cell and assume what's called proper position with these centrosomes lined up in the long direction of the cell. Now this is very important that this is done correctly, because the position of the mitotic spindle, if you mechanically moved it around, you could mechanically change the way in which the cell division takes place. So the position of this object when cell division is taking place, you can see these furrows that are starting to come down as cell division is heading towards you. It's really being determined by where these chromosomes are. And you need to have an asymmetric cell division so that you can have kind of variability in cell type later on down the road. Okay, but here's some of the elements of that. So the question is, it's known that the microtubules are very much involved in doing all of this. And the question is how? Exactly how is that happening? But here's some of the elements of what's being shown in this movie. There's pronuclear migration. I have the female and male pronuclei kind of fused together. They're going to form the mitotic spindle in a bit. This moves towards the center of the cell, and this object apparently rotates so that these nucleating sites are lined up. The nuclear membrane dissolves, and you form the mitotic spindle, and the chromosomes condense together onto what's called the metaphase plate. How they end up on a plate and flat and all that is obviously a force balance question, which is not understood. It's a really interesting question. And then the duplicated chromosomes are apparently pulled apart, and then you will have cell division soon thereafter. Okay, so these guys, These microtubules, these green sticks, are really what we're after. Okay, so how does that happen? Well, there's at least 3 mechanisms by which it's known that you can exert a force on a microtubule. One is because microtubules are polymerizing, they can actually exert forces through polymerization, right? Just like with cell crawling, when you can have an actin gel polymerizing up against a membrane, it can cause motion. And so the fact that you're polymerizing towards the surface means that you can push back from the surface by the polymerization force. I told you that dyneins are known to pull. They're actually coating the inside of the cell. And so I know we call that cortical pulling, pushing. And then there's a third, which is, as I told you, you can have dyneins coating organelles. And so imagine I have a free object here, which is, say, the pronucleus, and here's a sample microtubule. And a dynein is walking towards the minus end. So I should tell you that this nucleating site here, it's called the centrosome again, is full of minus ends, so to speak. So when it nucleates out a plus end. And so when you see things walking around along microtubules in these stages, they are walking towards these centrosomes, towards these nucleating sites. So if they're doing that, then they're using, if you like, they're using the cytoplasm or the fluid inside the cell as an anchor, it's a drag anchor. And so it's pulling up on this guy, pulling themselves down so it can actually move the attached object up. Okay? So there's 3 possibilities. And we've spent some time investigating all of these possibilities to see whether we could identify things that would be helpful to experiment or would somehow exclude them from consideration. And this is work of mine primarily with Ehsan Nazikdost, who's now at UNC Chapel Hill in their new applied physics department. And so here's these, I'm sorry, you can't really see this, so let me draw with my hands for a moment. So I'm going to show you 3 simulations. Each of those simulations is going to represent the pronuclear complex, this combined male and female pronucleus, as just a sphere, just a payload that's being dragged around. What it's being dragged around by are microtubules that are attached to centrosomes, which are attached to the nucleus. What you can see, because of the coloring, is that this is actually taking place in an ellipsoidal confinement, rather shaped like an embryo. Okay? And so it's a 3-dimensional simulation, and it's solving the Stokes equation using boundary integral methods. There's about 1,000 microtubules in this, and it's a big piece of code that's all bound. I'll say a little bit more about it, but it uses kind of very high-end ways of doing things. So fast kernel-independent fast multipole methods, kind of fancy time integration schemes, things like that. Kind of things I like to do sometimes. So so here's a model of pulling by payloads that are immersed into the cytoplasm. So here we start this thing off at the posterior end, which I can see very clearly on my screen. Oh, there it goes. You can now see where the boundary must be. So these guys, when they hit the boundary, they just depolymerize. But while they're not depolymerizing, we have a model for the action of things crawling along the microtubules and exerting forces equal and oppositely upon the microtubule and the fluid. And what you see from this is that you ended up, this is the long direction of the cell, which you cannot see, but I'll tell you that that direction right there where the centrosomes are lined up is the proper position for this. So this simulation, and I could explain some of the interesting aspects to it in terms of mechanics, will move this object into the center of the cell through this process of being pulled on by cytoplasmically bound dyneins, and then rotate. And that's really a torque instability that says, I want to have my centrosomes lined up with the long axis of the cell. That turns out to be the mechanically stable case. You can push. These are like the sentinel in Matrix 2, right? So it turns out that this will do the same thing. It takes a little longer in this particular simulation. You can make it faster by playing with parameters. There it's starting to rotate. One thing that you see from this is that because it's pushing, and we're assuming that it sticks to the cortex, it's constantly buckling or bending because it has a fixed end on the boundary. So, and the morphology of the microtubules is very different in that case. And here's one where you have cortically bound force generators, and you have a microtubule that has some probability of finding some site on the boundary. If it doesn't, it just depolymerizes. If it finds that site, it gets pulled. And again, nice centering, all these kinds of things. And so the disappointment in this is that with reasonable choices of parameters, using things that are known and guessing the rest, you can get all three of these mechanisms to do what they're supposed to be doing in the right timescales. Great. Okay. What that really means is theory's not helping you a lot, and you should get somebody to do some experiments for you. Okay, but one thing that does come out of that is, as I'm sure all of you have already read, this guy is like a force-free object. It's attached to objects that are force generators are attached to, so the whole thing is force-free. And so the flows that it generates inside of this cavity as it's moving are really something like a polar flow of a clammy or something like that. On the other hand, these two cases, they're using the boundary of the cell to push against, so they're transmitting force directly through the microtubules to the thing to which they're connected, so they're pushing that object around and absorbing the force on the boundary that it takes to do that. And so these look more like Stokeslets. They have different kind of flows that come out of that. So that's one thing. Okay, I'm not going to talk about the methods because I think I've chewed up quite a bit of time, except I'll say a few things. So I can't stop. Okay, so but what this is all based on is really work that I started a long time ago with Anna Karen Tornberg on building numerical methods for suspensions of flexible and extensible flexible fibers. And I wasn't thinking at the time about problems in cell biology. I was thinking more about non-Newtonian flows. And but basically, it's built on slender, non-local or local slender body theory, as was developed by Keller and Rubinow and by Johnson, where you can represent the flows induced by a skinny object moving through a fluid in terms of a distribution of Stokeslets along a center line. And we couple that, and we couple the flows produced by such objects. Oh, here we are. We assume that they're all inextensible elastic beams, and that's what the force looks like. And then we couple all of this into a boundary integral formulation for the induced flows from all the other surfaces. And so we use a boundary integral method for that, the so-called completed double layer formulation due to power of Miranda. There's many choices one could make, but this leads to nice second kind well-conditioned integral equations that you get to solve. And then this gets all put together within a parallelized FMM, fast multipole method network, or rather framework, with a preconditioned GMRes solve for the big linear solves that you have to make. The end result is that you end up with a scheme which is O of n per time step, where n is just the number of points you're using to resolve the fibers and the boundaries. It's not a volume solve. Okay. So, okay. So there's that. That's how we did all of that. So there has been some work very recently in trying to suss out what mechanisms are at play and what's generating forces and things like that. They're extremely rare experiments, and I just want to highlight one here, actually perhaps the one, as a matter of fact. And so there was a recent paper last year in Science from Joe Howard's group at Yale, and what they were able to do, well, they injected magnetic beads into the gonad of a parent worm. It got taken up by eggs. They would harvest the eggs, and then they would put this into a magnetic trap, and they could exert forces on the bead, and then they could look at the displacements. So this took a couple of postdocs, a couple of graduate students. This kind of thing. It's very, very, very tough experiments, but this is what they found and some of the conclusions that they reached. So they're trying to probe some of these mechanisms. So often what's talked about is what are the force mechanisms? I talked about transport, but there's a related question to once all this structure reaches the right place in the cell, how does it maintain its position? So that's what's called the centering problem. How do you stably maintain position? What are the forces that do that? Are they the same ones that caused it to migrate, or are they different forces? These kinds of things. And so this is what Joe's lab was looking at, was this one case where once you've got yourself into the right place of the cell, I just argued to you that theoretically we seem to be able to do that in different ways. Can you then probe that kind of more stable state and see what's causing what forces are doing things? So there's a magnetic bead, and so what they had a calibrated force, so they knew that they were exerting 16 piconewtons, for example, on this bead. They would maneuver it so that it was near one of these nucleating sites, so it could kind of nestle in all these microtubules and be kind of stuck there. They would apply a force to it, displace this, and then look at the relaxation or lack thereof. And so they modeled with the spring dash pop model. So here's the force on the bead. I have the bead with some drag coefficient, and I'm assuming a spring response. Very simple model. Because this is what you see out of the experiments. You turn on the force, it displaces, it relaxes, it displaces, it relaxes, it displaces, relaxes. So he's modeling with the dynamics. It looks rather like this. And so from that, they could calculate a relaxation time, which is somewhere between 20 up to 40 seconds, depending on the cell and how you do the fitting and things like that. They also did experiments where they used a mutant, an RNAi screen, it's called GPR12, that interferes with force generators on the boundary, with dyning at the boundary. And what they found was that if they knocked down the cortical dyning in this way, they had a stiffer spring response. So for the same force, it would actually stiffen. And so they interpreted that, the dynamics, as being a balance between pushing forces that were coming from polymerization and pulling forces from the boundary, and they were decreasing the pulling forces, and that's what was causing the stiffening, because I'm now pushing rather than pulling on the object. Okay. What I'm going to show you is that you can still get that kind of behavior even in a pure pulling model, and it's consistent with a set of new experiments. Okay, so here's how the experiments went, and this is high-end. So Dan has developed a very nice laser ablation setup where he can ablate microtubules. He can cut microtubules in 2D sections, 2D surfaces that can be pretty complex in shape. And so what's going on here, without substantial heating or damage, they claim. So me being a theorist, I'll just take that as he says it. And so here is a movie where they're just cycling, where high-end is cutting through this section of microtubules. So this is I should say, you may have noticed in some of the first movies that this mitotic spindle was rocking when it extended and you started to separate the chromosomes. So this is during that stage. It's called spindle oscillation. And she's cutting as this is moving up right there. You can see the microtubules depolymerize in both directions. And you can also see, if you stare close enough, that right there, it kind of accelerates down a little bit. So you need to quantify those kinds of things. Okay, so what they did is focus on this oscillation stage. So I'm going to kind of move backwards in time. I think I have another 10 minutes.
But that includes questions.
Oh, okay. All right. So I'm going to move backwards in time, starting with this kind of oscillation part, because it's so transparent what's going on. So here's oscillation. And so high-end is tracking this kind of center of mass of these bright spots as it's oscillating. This is the anterior pole, that's the posterior pole. And you see you have this kind of burst of oscillation as the spindle is elongating. That doesn't always happen in C. elegans, but it almost always happens. And you have a very characteristic time scale, frequency, that pops out of this all the time. So the idea is to do laser ablation during various subcycles and positions in time in this kind of oscillatory dynamics. So when it's at the midplane, when it's at the peak of its oscillation, things like that. When it's at the midplane, you may cut above, you may cut below, and you figure out whether something's being pushed or pulled. Okay, so here's the first experiment. So they take the posterior pole, it's moving down, it's going through the midplane, and what they do is they cut, as it's moving down, all the microtubules below. The response is that I don't know if you can see this, but what happens is that these are the controls, and the cut ones rebound up, as though it's being pulled. And I've cut all the ropes that are pulling it down, now it rebounds up. On the other hand, if you do the same time, but now you cut above, it changes a little bit. It goes a little bit more deeply, but not much. It doesn't perturb the dynamics very much. Seems to be saying pulling forces are predominant here. You're being pulled and you're cutting the pulling things. And there's an oscillation in who's pulling on you. Are you being pulled from above or below? Now you cut it at an extreme. You've reached the top of your amplitude and you're about to reverse. And then what you do is you cut down here. So here you're in zero force balance, right? And presumably if it's pulling, this will start to increase and pull you down. And indeed, that seems to be what it's saying. Here's the controls again, but the ones that are cut, it stalls or even rebounds back up towards the cortex. And again, you get a similar thing that if you're at the peak and you cut here, you have kind of small forces in balance is the idea at this point, and then it has very little effect. And then they do much more sophisticated cuts to rule out various other kinds of things, like what's going on in interactions, just not above and below, but from the sides and so forth. Okay, so then there's another question. Seems to be pulling. We have two mechanisms for pulling, we think. We have pulling from cytoplasmically kind of floating dyneins, or those that are sitting upon the cortex. And it turns out that from our modeling, we really think that those should have two different kind of fluid mechanical signatures. And so if I'm pulling like that, then I don't have any sort of active forces acting on these microtubules. And so if there's some motion of these microtubules, then the fluid should just be kind of dragged around with it. On the other hand, if it's associated with pulling by things attached to it, then there will be fluid flows that are in the frame of the microtubule, and they should all move along with the microtubule. That's the idea. So here, for example, is a simulation of the cortical pulling mechanism when you're in an oscillation. And so what you see is, because these are now just no-slip things, there's no active force acting upon them, then the whole thing just, if you rotate it by a force, then it just drags all the fluid as kind of a rotation. And on the other hand, if you have force generators that are dragging fluid around in here, you may even be rotating it, but you see these very large flows that are associated with these motors moving along. So they did another heroic experiment. So they injected nanodiamonds into the gonad of a parent worm, and they were able to do PIV on this kind of 50 micron, 35 micron thing. They do this over many cells. So the PIV, the nanodiamonds are these bright spots. There you are going through elongation and this kind of stuff. They do PIV, and then they average over many such cases. And what they find is it looks a lot like basically what would be consistent with pulling from the cortex and the rotations associated with that kind of mechanism. Okay, so it seems to be that in that stage we would conclude that cortical pulling is dominating, and that's the thing that's really associated with spindle oscillations. So then the question is, where do these spindle oscillations come from if it's a cortical pulling mechanism? So we have there's been some models of this. This is our own model, which is consistent with a lot of data. And I guess I should stop soon, which I will, maybe. So I'll just kind of outline the model. It's more of a proof of concept model because it's not like a full fluid mechanical model as yet. Kind of heading there now. But the basic picture is that you have a nucleating center, a centrosome, and microtubules nucleate there. They grow at some constant velocity to the boundary. If they don't find a force generator, they just depolymerize. If they do, they attach to it, and they have some different detachment rate. So those that hit and bind a motor experience a pulling force. Okay. Those that don't, they depolymerize. Each force generator holds one microtubule, which means this is a model where you don't have a large number of force generators, but rather some sparser covering, and microtubules compete for it. And that seems to be important. And then we just have a simple model where we have a drag coefficient and forces some to drag the centrosomes around. So here's how such a model works. This is, again, not a full fluid flow, but more of a stochastic simple drag model in a geometry. And there it is, where what's happened is that we've increased the number of force generators on one side, because it's known that's what happens as you move into oscillations. And this will spontaneously go into oscillations, and there it is. The red ones that you see here in these microtubules, we have about the right numbers. The red ones are the ones that become attached to the boundary to force generators. And so how does this work? I can describe it. So we have a 1D kinetic model that actually kind of, I think, susses it out pretty well. But basically, if you have enough pulling force, then you can make the centrosome, you can pull it faster than the polymerization speed. What that means, if you end up pulling this whole object away because you have some imbalance that pulls you up, then you detach microtubules down here because they can't grow fast enough to be recaptured by force generators because the speed is greater than the polymerization speed. And so as it moves up, because it has a finite number of these force generators up here, the ones that are pulling you up, as they get closer and closer, they see fewer and fewer force generators because of the geometry. So it slows down. As it slows down, that gives this enough time to build up and to start to pull you back, and the whole cycle just goes. And so that actually predicts a frequency that scales very naturally on the growth velocity divided by the scale of the cell. And Hyen did some knockdown experiments, again, GPR12 knockdown experiments, and you can't see this very well, I'm sorry. But basically, what the knockdown experiments show you is that as you decrease the number of force generators, so you think in the experiment, you keep a fixed frequency, you vary the amplitude, but you have a linear relation that's telling you you have a fixed frequency. Okay. So our model does all of these things. So in that same kind of knockdown experiment, we change the number of possible force generators in the boundary. It causes a behavior like this, where the oscillations will stop once you get down on the order, once the oscillation speed gets small enough that you're on the growth velocity speed. So here it's cut off in the experiment. We find that too. It's just very natural in this type of model. And I don't have time to talk about this. Base battery is low. I may not have much time at all. But we can also reproduce this stiffening of springs that's associated with this geometric effect of having a kind of a sparse number of force generators. And so that may force me to a conclusion. Oh, one other thing it does, and this was a prediction that came out of our model that they then went and confirmed, is that because it's a microtubule nucleation model where you're constantly nucleating microtubules, and if they hit the boundary, they depolymerize, and then you wait for them to nucleate again. What that gives you is a cycle in the number of microtubules that are in the system as a function of how far this centrosome is from the boundary. There's the dynamics associated with that. And so if we measure it in our experiment, we see this variation in time. And this would be, say, the top or bottom microtubules. The ones on the side don't change much. And this is, you're not seeing the error bars here. There's fluctuations in this. It looks a lot neater than our data. But if you look in the background, all the samples, it's kind of messy. But the point is you see that same kind of variation they found then when they went and looked at the intensity of luminescence in the experiments. Okay. So I'm going to stop there, I think, because I'm running out of time. I have run out of time, except to say that he's gone back and looked at laser ablation in all the earlier stages of all of this. In metaphase, where it's holding place, in migration, all these kinds of things. He absolutely destroyed the cytoplasmic pulling model by a very beautiful laser cut experiment that discriminated very cleanly. So it seems to be pulling forces all the way down. And I won't talk about that, and I'll just give you a summary. So what we're after now is we want to build this kind of knowledge in kind of a soup to nut simulation, including fluid mechanics. There's some things in where we don't match that we think have to do with drag coefficients changing as things get closer to boundaries and that kind of stuff. We do have a one-dimensional kinetic model that reproduces in kind of sketch the things I talked about today. And it brought up another question about whether this type of picture of surface-bound force generators might explain other phenomena, and the answer seems to be yes. And thank you. I'll just play this in the background because it's pretty. And take any questions I have time to take.
Let's have maybe one question.
Severio.
Oh, yes, except from Severio, because you've got to come up here
Yeah, that's true.
and make a talk.
Ask me on the way.
I have a question, but it's a bio question. I hope it's okay.
Yeah.
What happens to the cell division if this positioning doesn't
work
you end
the way
up,
it's supposed to
you
work.
may go into a cell division, you may end up with a non-viable proliferation of cells
You
after
said at the
that.
beginning that it's important to have variability.
So my understanding is that if you can position the spindle right into the center of the cell, so you have more or less two identical cells, you will not produce a viable organism because you don't have variation in cell type.
Okay, thanks, Mike.
Yep.